Ready for coming. I'm Rachel Green, and I do scientific customer support for the RCSB. I'm originally a crystallographer by training, but right now I love to take questions from users and help users. So just to start, want to say I'm going to give a little introduction. We're going to talk about the structure summary pages today, which really focus on individual structures. And so we'll do a little introduction and then I'll open it up to questions. But if you have a question that doesn't get answered during this time, or you think of it later, or you feel like it's kind of off topic, some other topic, whatever it is, you can hit the contact us button on the home page, or really any page on the website, and you can fill out this form and submit it. And I'll be happy to answer your question, whatever that is. So again, that's always going to be up here at the top. So just to start, I'm going to show you kind of how to get to the structure summary pages. And there's a couple of ways to get there. One is just by typing an ID up here in the search bar at the top. Our usual favorite is 4-HHB, and then you can hit return and go directly there. You can come down here, and we're going to actually look at we have the latest entries. This is updated weekly. We're going to start by looking at one of the entries in here. You can also do a search. We'll come back there in 1 second. Whatever kind of search you want to do from the top bar, and then you get a list of structures. And if you click anywhere in here, you're going to get to the structure summary page. Okay, so just going back to the home page again, we're going to click, let's see. Actually, I'm just going to go straight there. This is a structure that was in this week's release, 9 JKV. And you'll see we also have the extended PDB ID listed here, which as we start running out of the 4 character codes, we're going to be moving to. So this is a structure summary page for this entry, and there's lots and lots of information on this page. To start with, there are a series of tabs along the top, and there you can click right to them, or within this page, there are ways to get to those tabs for the additional information. So for example, to look at the structure 3-dimensionally and manipulate it, this is a program called Molestar. You can use that tab, or from here, you can also click there. So these tabs offer lots of different types of information. This is the annotations tab. So anytime you see something in this color orange on the website, it's going to indicate that the information is coming from an external resource. So this is all information that is collected here for you about this entry from external sources. This will tell you information about the experiment. This is the sequence browser, which we're actually going to come back to this in a second from the main structure page. But just coming back to here now, I want to show you if you would like to download files. This is the download file menu. You'll see here that there's the MMSIF format available. For this entry, there is not a legacy PDB format available. We're moving away from the legacy PDB file format to the MMSIF format, which is the current archive format. So for this particular entry, you cannot download a PDB file, but you can download some validation information and so on. This happens to be, okay, let's see. Oh, this was actually not a great example because, well, okay, I was going to show you some of the literature, but this is still to be published, which is fine. So just back up to the top. There's lots of things on this page that will launch an additional search. So for example, here, this structure is from the human organism. And if you click this link, it actually does a search for all the structures from that organism. So there's lots of places on this page where you could do that. You can do that with authors. You can do that with this classification. Um, so lots of places where you could do that kind of thing. So moving down, we have here is the validation slider, which gives you an idea of the quality. You can click on full report and see this in more detail. Over here, you can explore the structure in 3D in different ways. For example, we'll just look real quickly at the sequence annotation view, which allows you to compare really the sequence with the structure simultaneously. So this is a really useful browser. And this is again the Molstar viewer, and you could manipulate this view in different ways. So continuing on, again, this is kind of a neat thing. You can do a search for similar assemblies right from here. So that will just bring up a search for that. And similarly, but not exactly the same, as you could also down here where you could search by 3D structure. So that's going to give you a search just based on the different parts of the structure. So down here, the structure is broken up into the different entities. So an entity is a chemically unique part of the structure. So you might think of it as a chain, although it's not always exactly correct, but you can think of it that way. So this molecule is in this entity, and this molecule is in a separate entity. There's lots of things you could do within each entity. You can do some searches by sequence. You can explore this. See if we click here. This gives you a view of different entries that are grouped in the same seat with the same sequence. Let's see, what else do I want to show you? This again, you can do some searching by gene name right from here. You could go directly to the UniProt entry for this sequence. I'm sort of throwing a lot at you here, and we could go back to anything that you want to explore further. So this is the sequence view, which is the same thing that you see up here in the tabs. And so there's lots of different tracks along here, and this is you can interact with this, expand it, and so you can look at the secondary structure. And if you see up here on the top right, there's some annotation of what you're hovering over. So this here is a helical region. There's just lots of information about disorder, where disulfide bridges are, really getting into the nitty gritty of this sequence. If you expand really far, you can actually see the primary sequence of the structure compared with what is in the UniProt database. And let's see, what else do I want to show you real briefly? So again, the entities are all listed here. And then you get to the small molecules section, which tells you about what ligands there are in the structure. And you can look for the same, again, you could do a query for the same ligand in other structures. You could, this takes you directly to a focus on this ligand within the structure, and you could look at the interactions there. I don't know why it keeps zooming out. So, and so if there's a lot of ligands in the structure, they'll all be listed here. And then down here, there's some more information about the experimental data, and this will take you to the experimental tab from above. And then finally down here, there's some information about the entry history and details about the revision, if there's been any revisions. And so you could click here and this will take you to this versions tab, and where it'll give you information about kind of the history of the entry and what's happened to it. So that's kind of a quick walkthrough of the structure summary page. There's a lot more there, but I think let's open it up to questions, and I'm happy to walk you through whatever I said that was confusing or that you'd like more detail on.